Welcome back, students. In this video, I am going to talk about dihybrid cross. The word di means two. So here the Mendel has selected two characters to study at a time. So when we are studying two characters at a time, we need to make certain assumptions. And it is the first assumption is that the plants selected by the Mendel are pure lines. It means they are homozygous in nature. And second thing is each character is governed by different genes and they are present on different chromosomes. Suppose if we are talking about this seed shape, if this is present on the chromosome 1 and this second character, that is seed color, so this is present on the chromosome 2. It means these two characters are present on different chromosomes and are governed by different genes. So these assumptions we need to make before studying the dihybrid cross. So the Mendel has selected seed shape and the seed color. So the first plant has the round shaped seeds. We are representing it as capital R, and the second plant has wrinkled seeds. We are representing it as small r. And the seed color, it is yellow, is the dominant one. So we are writing it as capital Y. And the green is the recessive one. We are writing as small y. So now let us look at the experiment. So Mendel has selected round yellow and green wrinkled plant. So this round yellow, it will produce the gamete capital R, capital Y. So it's very simple. So if you see this capital R, both are same. So you need to write one. And this small y, both are same. So you need to write one. So multiply them. 1 into 1 is 1. So only one type of gamete is produced. That is capital R, capital Y. So here also the same way. Small r, write 1. And small y, write 1. Multiply them. So the answer is 1. So here also only one type of gamete is produced, that is small r, small y. So when these two gametes fuse, it will result in capital R, capital Y, small y. As capital R is dominant over small r, it will be round in shape, and capital Y is dominant over small y, it is yellow in shape. This is in accordance with the law of dominance. So for producing F2 generation, so we are going to fuse this capital R, small r, capital Y, small y, that is F1 cross F1. Okay, so we are going to cross F1 with another F1. So if you see here, this capital R, small r, both are different. So you need to write 2. Here capital Y, small y, both are different. So again you need to write 2. So multiply them. 2 into 2 is equal to 4. It means 4 different gametes are produced in this case. So what are they? Capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. So this is how 4 different gametes are produced from the plant. So as there are 4 different gametic combinations in both the case, so 4 into 4, it will give rise to the 16 different genetic combinations. Okay, so we are writing the gametes of this plant on y-axis and gametes from this on x-axis, and we are drawing the checkerboard. So if you see here, there are 16 different combinations. So of these 16 different combinations, the straight line over here, this diagonal, and this straight line. So all these 9, if you count them, there will be 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. So these 9 are round and yellow. And this one, which are represented in rectangle shape, 2 and 3. So these 3 are round and green. So you can see in all these cases there is small y, small y. So it is round and green. So these 3, you can see all are smaller, smaller. It means wrinkled, and there is at least one capital Y in all these 3. You can see both of them are capital Y. Here one capital Y is there. Here also one capital Y is there. So these 3 are wrinkled yellow. And this last one, this is having completely smaller, smaller, and small y. This is one wrinkled green. So out of these 16 combinations, 9 are round yellow, 9 are round green, 3 are wrinkled yellow, and 1 is wrinkled green. So if you see the genotypic and phenotypic ratios, the phenotypic ratio is 9:3:3:3:1, whereas the genotypic ratio is 1:2:1, 2:4:2, and 1:2:1. So these are the phenotypic and genotypic ratios of dihybrid cross. So if you want to derive the genotypic ratio of dihybrid cross on your own, it's very simple to do it. So what is the genotypic ratio of monohybrid cross? 1:2:1, right? And what is this one corresponds to? Let us assume capital R, capital R. So 1, 2, 1. So 1 capital R, 2 capital R, small r, and 1 small r, small r. So subdivide this into 3. So write what is the monohybrid genotypic ratio? 1, 1, 2, and 1. 1, 2, and 1. 1, 2, and 1. So what is the second character here? Seed color, that is capital Y, capital Y, capital Y, small y, small y, small y. Write the same thing here, capital Y, capital Y, capital Y, small y, and small y, small y. Capital Y, capital Y, capital Y, small y, and small y, small y. So now multiply them. So 1 into 1 is 1, capital R, capital R, capital Y, capital Y. 1 into 2, 2, capital R, capital R, capital Y, small y. 1 into 1, 1, capital R, small y, small y. 2 into 1, 2, capital R, small r, capital Y, capital Y. 2 into 2, 4, capital R, small r, capital Y, small y. 2 into 1, 2, capital R, small y, small y. 1 into 1, 1, small r, capital Y, capital Y. 1 into 2, 2, small r, capital Y, small y. 1 into 1, 1, small r, small y, small y. So now add them all. So 1 plus 2, 3, 2 plus 1, 1 plus 2, 3, 3 plus 1, 4, 4 plus 2, 6, 6 plus 4, 10, 10 plus 2, 12, 12 plus 1, 13, 13 plus 2, 15, and 15 plus 1, 16. Right? So what is the ratio? If you write in a straight line, 1 is to 2 is to 1, 2 is to 4 is to 2, and 1 is to 2 is to 1. So this is the genotypic ratio for dihybrid cross. So based on this, Mendel has proposed law of independent assortment. So what is saying here? Both the characters are transferred in 3:1 phenotypic ratio. Both the characters are transferred in 3:1 phenotypic ratio or 1:2:1 genotypic ratio. So this he called it as law of independent assortment. Okay. So if you see in a plant, within a plant, these both the characters we are studying simultaneously, but while getting transferred from one generation to the next, so this is also following the 3:1 pattern, and this is also following 3:1 pattern. So this character, even though it is going in association with this character, they are not getting influenced in any way. There is no domination or any other influence in any way. So that is what he is explaining in law of independent assortment. So this states that when alleles of a two different characters, when alleles of a two different characters are present together, the segregation of one pair of alleles does not influence the other pair. So this is getting inherited in 3:1 ratio. So this is also getting inherited in the 3:1 ratio. So this is called as law of independent assortment. So try to make a note of these questions, write answers on your own. Thank you for watching the video. In the next video, I will be talking about the sex determination in humans. Like and share the video with your friends.